glioblastoma. If this one data set being all sorts of or different molecules in the and based on this and these different data types is going to and one glioblastoma methylation a lot of data types each sample separate like dimensions of a cell. One way to sort of organize this all together, so be pretty familiar. We've got a genome that leads to a messenger RNA, which leads to a protein, and then some sort of biological function that results from that. And what I'm showing here is a factor graph, which is a sort of a general graphical model. It's a lot like a Bayesian network. So each variable here is a circle. And we're using 3 states discrete variables here. So relative to some normal tissue, each state describes whether in the cancer sample, whether it's up, down, or the same. And in between each of these different variables is something called a factor. In a Bayesian network, this would be a conditional probability table. It's just a list of, it's a function that takes this, the 2 variables and gives you a number back, basically. And so with all these different types of data, we can now sort of map them onto this sort of central dogma. And if we have array CGH or other sorts of copy number information, we can attach it to the gene copy number hidden node. And if we have transcriptomics, we can fit it over there. And as we get more and more types of data, we might have some information about what's going on at the later stages of the central dogma. But one key part of all this is that right here, there's a lot of regulation going on between having a gene in the genome and having a messenger RNA result from that gene. So this also happens all the other stages in between the messenger RNA to the protein, and also just general activity. So we need to include this information when we're thinking about how all this data comes together into one big picture. We need to know what is the transcriptional regulation that is controlling the difference between these 2 hidden nodes. And particularly in cancer, we know that signaling is a big part of it, and we need to sort of be able to incorporate what's going on with all the signaling. And especially in cancer, this is important because it's been known for a long time that it's not a single gene that causes cancers. There's many different, it's not one particular gene in a pathway. That's a problem. This is a issue from TCGA in 2000. And what they found when they did sort of an exhaustive p53 signaling is perturbed in about 90%. But if it's just one single data type or one single gene, you're not going to find data type. p53 is perturbed in all these. So the gene CDKN2A is highly perturbed, and this is seen, but still doesn't explain all of your samples. So you have to see what's going on in order to find out how the pathway is perturbed. And the inference matters a lot too. So knowing that MDM2 is an inhibitor of telling you that it's probably going to be amplified rather than deleted. So science can tell a lot about what type of fusions to expect. Okay, so back to that little central dogma picture. I'm showing you a little small snippet of the central dogma for 2 different genes here on the left. So there's a transcription factor from the genome down to activity. We can hook up the transcription factor's activity to this regulation interaction between the DNA and the mRNA in its target gene. Similarly, for post-translational modification, we can hook up the activity of a kinase to the regulation of activity of the phosphorylated gene. So if the kinase activates a gene, increased activity here will result in increased activity there. If the kinase deactivates a gene, we'll see exactly the opposite effect there. There's a few other very common features and pathways that would be nice to model as well. Protein complexes often play a role in cancer, so we want to be able to sort of say to summarize what's going on with the complex. We won't get actual data on a complex, so we'll get what we infer all these activities, and then we can infer sort of a general complex activity after that. Similarly, gene families are often annotated in a pathway where you have many different genes which have nearly identical function, and any one of them will be able to provide that function. So the topologies of these two are identical. What's different are the little black boxes, the factors. And we're modeling parameters for these factors in two different ways. So for a protein complex, we have an AND function. So we want to we need all of the components of the complex for the complex to function. And for a gene family, we have a noisy OR function. So any one of the input genes is enough for the gene family to function. Another section of parameters are the interaction parameters, the regulatory parameters. And so we're taking a lot of these from an actual pathway. So looking at, from a curated pathway structure. So for example, we might know about 3 different transcription factors for a gene. And we know that maybe 2 are activators, 1's a repressor. And we're modeling this with a weighted, with a noisy vote of repressors and activators to say what the mRNA state should be. And the final set of parameters in the model that I haven't told you about yet are the observation parameters. So going back to wherever we hook in data, we can learn these through expectation maximization, so simple standard procedure in graphical models. And going back to the big picture of it all, what we're doing is we're starting with a picture and go out into a graphical model into the stage where we might want to we might have these abstract nodes that we don't have direct data. What I'm showing here is that perhaps this in one sample in the container of DNA, and that might translate more activity of M2, which in turn degrades the results in the post. So in one particular sample, this might be what happened. The for us is something, and for each gene, what we're doing is we're looking at just this last, or for our process, we're looking at its one factor graph, and we want to calculate its activity. And we start with the 3 possible variables. So up, down, as normal tissue, and we calculate the log basically the log odds minus the prior. And for the different states, we get a few numbers, and then a single number, just the maximum, and then multiplying it by the size is the largest log ratio. We have one single that says that gene for in the general setup of data pathways with and I'll show you results free and currently we're pathway data get top and no pathways. As one of this starting here plot both data and our inferred quantities, the IPAs. And the way to read this diagram is that each spoke on the wheel is a different sample. So here there's one sample which has an increased copy number, increased expression, and we're inferring a high integrated pathway activity for it, and then I'm also showing the estrogen receptor status at the bottom. And the reason we draw it like this is so that we can show the rest of the pathway as well, or at least a section of it. And if you look at AKT1, which is generally highly expressed, and we infer it to be very active, we can see that the sections where it is highly active, the same samples are also having decreased apoptosis. Or we're inferring that it's decreased apoptosis within this single pathway. Now there's one other regulator of apoptosis in this pathway, and that's this complex, which again we don't have direct data for, but we're combining the data from other components. And so for example, none of these other, in this little section there, NRG2 is not particularly expressed, or increased copy number, and also ERBB2 and 3 are about at the same level. And so for the complex, we're inferring in that little section that it is not extremely activated. Now for NRG2, it's seen extremely high expression, and that results in having a very slight increase in the complex, the chance that the complex might be active. This isn't, however, it's hard to tell from the colors, but this isn't significant. But fortunately, we have real numbers behind this, and I can show you the comparisons. Here's a different pathway, and to sort of assess the significance and tell which of these IPAs are important based on value, we create null data by taking a tuple of genomes we have and assigning that to a null patient and randoming the data. And we calculate these gray boxes, which are sort of the back distribution of an IPA in that pathway. So this sort of takes into account the pathway structure, so to help normalize the pathway structure. So A1 is also in this other pathway, class 1 PI3K signaling, and it's also the most activated entity of that pathway. We have 6 of the most activated entities, and some of these are abs nodes. So we're saying that, we're predicting that G1S transition is up in these samples. Also, we're saying that MDM2 is up. However, the comp that MDM2 forms is not up, so it's not significantly changed at all. And this is because the other components of that complex are not significantly changed. And at the other end of the scale, we have negative regulation of cell cycle, which we're predicting to be significantly down, which sort of the brakes have been taken off of the cell cycle. So what we get here is that there's lots of, we can assess the significance of a pathway, or of an end of a pathway, and also the pathway itself. So we can say whether this pathway has been significantly converted on average over patients. And this is important because which pathways are meaningful or actionable. Maybe a pathway that describes lung regulation won't be whole. So we want to be able to distinguish between pathways that may be important or pathways which are either, might not be well curated or might not be complete. And the way we can sort of get at that a little bit is to compare an aggregation of pathways. So this is more where we have taken the same pathway structure to this pathway structure. So we created a bunch of pathway databases and or real pathway structures on the variables. 2 lines here are the times basically a rock plot. I'm sure results from 2 different sets, one is cancer and it's hard. It was something called SPEED, which uses the pathway structure pathways, which only operates on expression data. He saw significant improvement in also pathway descriptive of. So there's reasons that we wouldn't get 100%. In glioblastoma, with different CGA, we were able to use types. So on the right side, here's cluster 4 in the and also only 4 in the other is hyper. Indicate that these samples are less aggressively. We compare the survival of there's or significant. So we're thinking when data, we weren't able to come up with IPAs. We think that there's some from the data or from the copy. this in a cancer data from TC. So it's all high grades on the face of it to say these. We were able to sort of put one cluster has a better prognosis compared to them. What we're doing is we're taking different data, we're taking pathway structures and combining together to get sort of what we hope is a unified view of all of it at once for individual genes within a pathway. And we're finding so far that by using this method, we're getting better results than we would if we just used copy number or expression data or any of the input data. There were a lot of people that helped with this. Steve mentioned before was the co-first author, and also the other cancer genomics people at UC. Thank the TCGA for providing us with data that we could, and also for a travel fellowship that allowed me to be listening.
About the multi-parameter approach to a better. One is it is often the case in gene we call it in pathway are not different message or protein. The question is have you model? My question is well in different types of certain cancers. A lot of cross proteins with interact and you're flexible enough to incorporate.
I'll have to ask you again about the expressions.
The first question is that many of the pathway are not actually expressed at the level of the message.
So to answer that one first, we're hoping that by the time we'll be able to catch up on the lesion. So it's not mutated, and there's no change in able to catch it. So we have some sort of data that would tell us what's going on. But we by incorporating the mutations and that we'll be able to do something on. As far as we're only looking at traffic, and so we to sort of to be able to learn more about this, but we out structures before we went much.
It's really interesting. One of the things I like about it is looking subtypes. Did you go backwards through the model, figure out what was it the activity protein level? Was it so you can go backwards and tell us what marker?
So a lot of time, it's most of our signals and expression. There's also expression. We're getting a little bit from halfway and say, copper change here to here, but it looks like most so far.
Also, the issue that quantities may not translate easily to level. Some things has hundreds that more potent.
Yeah.
Right.
And that is a problem. One way we kind of hope to deal with that is we're teamable. So all measurements are sort of also we're for activity compared to gene. And so each gene will have a different range of importance. I mean, as with gene expression now on the face of research.
I've actually one more technical question. It's often a pathway. How this new phenomena?
If a gene appears to have different activity in the context of each pathway. So it's really dependent upon the particular pathway frame of knowledge. Through we have activities because P53 and so far when we've looked at the pathways have matched up pretty well. We find I'm sure that we will have different paths, and then we'll have to sort of chase it down and see what.